So good morning, everybody. I'm short. I have to do this. Good morning. So, as Brad was mentioning and thinking about the timeline that he has put up there, it has been 11, 12 years now that you and I have been working together, even since before the start of the FTD clinic. And it's really nice to be such a member of a team that works so hard, and we're making progress slowly but surely. So I will be discussing the genetics of FTD, just giving a brief overview, and then we have a session this afternoon also. I usually start with a little biology lesson to make sure we're all on the same page before I talk about FTD specifically. So I wish this pie would change. It hasn't really changed much over the last few years. Trying to find the genes that are associated with FTD. As you can see on the left-hand side, most cases of FTD are sporadic with little or unknown family history. And for these folks, we don't have any genetic testing available. It's typically due to multiple genetic factors as well as life and environmental factors, and why it typically comes on later in life for those folks with FTD. At the top right there in yellow, we have families who have strong family history, typical multiple generations with multiple family members, males and females, that have FTD. And for the majority of those folks, we can find a genetic alteration that is occurring for them. But there's still lots of families that we cannot find a genetic alteration for. So we know we have more that we need to identify, and that's part of our research. And then in green there, we have families with FTD and maybe some other neurodegenerative conditions, whether it's Alzheimer's or ALS or Parkinson's. We always certainly question what the diagnosis is, especially when someone doesn't have an autopsy. And for a proportion of those, we can find a gene mutation also, but for the majority of them, we are unable to. So we certainly have lots of work left ahead of us. There's a very small piece of the pie there on the bottom right for folks that are the only individual in the family that have FTD, and for a very small proportion of those, we do find a genetic alteration. And these are typically folks who are very, very young, and a spontaneous alteration occurred in the pregnancy that formed them. Okay. So as I mentioned, just a little biology lesson to make sure we're all on the same page. I know there's folks here that have been here previously or I have met with, but we have new faces too. So we start off with DNA. That's the genetic molecule that we hear so much about. This is how we carry our information. DNA is organized into thousands or millions of units. Building blocks of DNA make genes. Humans have about 20,000 genes. And I think it's important to note that we all have the same genes. They just have some variation, so that's why we all don't look or act the same. And I think this is incorrectly reported in the media at times. They say, oh, so-and-so has this gene, or folks who have this condition have this gene. We all have those genes. The genes I will be discussing in a few minutes, we all have and we all want. So I think that's important to note. It's when these genes don't work as they should that they can cause some medical concerns, okay? So Angelina Jolie was the most recent one in the media, and they say, oh, she has the Brca gene. We all have the Brca gene. It is a tumor suppressor gene. So as its name implies, it stops tumors from growing. For Angelina Jolie and her family members, it doesn't work correctly, and that's why they have an increased risk for cancer. Okay, so again, the genes that I'll be discussing that are associated with FTD, we all have, and they're all important for normal brain function. It's when they don't work correctly that we can have some concerns. Genes are housed on structures called chromosomes in our cells, and humans have 46 chromosomes, 23 pairs. So that's a nice colorful picture there on the right of what our human chromosomes look like. How do we get our genetic information? Well, we get it from mom and dad, the good and the bad, right? That's how we are who we are. And how does this occur? So as I mentioned, most cells contain 46 chromosomes, 23 pairs, and that includes immature egg and sperm cells. But egg and sperm cells do something special so that we can reproduce. The chromosome pairs separate and the cell divides in half. So this is a picture of immature egg and sperm up the top here. They're just showing 2 pairs instead of all 23, so it's not as cumbersome. And when the egg or the sperm matures so that it can be used for fertilization, the chromosome pairs are going to separate and divide in half, so that a mature egg only has one copy of each chromosome, as does a mature sperm. So when they come together, the baby has 46 chromosomes, 23 pairs, just as we're supposed to. If we didn't do this, we'd have too much genetic information, and that's not good. Okay? So this is how we're half mom and half dad, and this is how we form a human. It's one single cell, an egg and a sperm come together, combine the genetic information, and we all start off as a single cell. And this cell replicates itself, all the genetic information and all the machinery, and then we get 2 cells, and then we get 4, and then we get 8, and so forth and so on, until we get a couple 100 trillion, and we have a human. So all cells have the exact same genetic information. So how does some know to be a skin cell, or a cardiac cell, or a neuronal cell? It depends upon what genes are turned on or off. So think of cells that start off, early as having the potential to become anything. They're clay that we need to mold, marble that we need to chisel. And then depending upon which genes we turn on or off, we'll decide what pathway they go down to. They're going to become blood cells, skin cells, bone cells, brain cells, all right? And it's genetics that's going to sculpt this. And this will be important when I mention those stem cells that we create, as Dr. Dickerson mentioned. So this is the picture that maybe some of you have seen me draw for you. This is a pedigree that we geneticists use. Squares are males and circles are females. And we diagrammatically show relationships, a couple, offspring, siblings. Folks who are colored in green here have FTD. And this is a typical picture we would see for one of our hereditary forms of FTD, multiple generations, both males and females. It is a dominant condition. You've probably heard that word. You only need one copy of our gene pair to be altered in order for symptoms to occur. And so FTD is inherited through these rare families in an autosomal dominant fashion. And because we know that it's dominant, the chance for offspring to also have the condition is 50%. And how do we know that? Well, it goes back to what happens with eggs and sperm so that we can use them for fertilization. Remember, we all have 2 copies of each chromosome and therefore 2 copies of each gene. And these chromosome pairs are going to divide in half to make our mature eggs and sperm. And so which copy of the chromosome, and therefore which copy of that gene, are you going to receive? And it's random, just like flipping a coin, heads or tails. So in this picture, here's Dad, who has FTD, and he has one copy of one of the genes that's associated with FTD that works just fine. Unfortunately, his other copy of the gene is altered and not working as it should, and that's represented by the capital D. And that is what we know is causing FTD for him. Mom is fine. She has 2 normal functioning copies of the gene. And remember, this is random. How is the egg or the sperm going to mature so that it can be used for fertilization? Therefore, what do we pass on to our offspring? And so if Dad has one functioning copy of whatever gene we're discussing here and one non-functioning or altered copy, you can see that 1 out of 2, 50-50 chance that occurs with each pregnancy. Folks sometimes think, well, if I have 8 children, 4 are going to have it and 4 are not going to have it. And that's not the way it works. Think about flipping a coin. You flip a coin, you get heads. The next time you flip a coin, you can get heads or tails. It's still 50-50, so forth and so on. So we certainly see families where there's 8 offspring and 1 of them have it. Or unfortunately, there's 3 offspring and all 3 of them have it. So this is completely random. It's part of our humanness. We're not machines. Okay. So we're trying to figure out what's going on and learn why folks have FTD and what is the chance for other family members. So we're trying to look at the genes that are associated with these rare hereditary forms so they can tell us information about the more common sporadic forms. So there's several genes that have been associated with FTD. These ones up here, VCP, CHAMP, FUS, DBP. They're very rare. I don't frequently test folks for those. I start off with the 3 most common ones that I'll give you a brief overview. Certainly we go forward with testing of these genes for some folks, but they have a very, very low yield for any information. So progranulin is a gene that's associated with the hereditary form of FTD. Remember, genes are housed on chromosomes, so this is just telling us its geographic location. There's lots of different alterations that can occur in the progranulin gene that cause FTD. Some diseases, everybody has the exact same change in the gene, but not for progranulin-associated FTD. We know the normal protein stimulates cell growth and survival. And unfortunately, what happens in FTD when this gene is changed, the protein, that's what genes do, they make proteins, is lost. So folks who have progranulin mutations have a loss of progranulin, and then we lose our ability to stimulate cell proliferation and survival. So because we can understand the underlying genetic cause and how this occurs, we know that what we need to do is replace progranulin for folks who have progranulin mutations. Maybe accounts for 5 to 10% of all FTD and maybe a quarter of familial. Penetrance means if you have a genetic change, what's the chance you will have a disease? And it's greater than 95%. It's certainly variable, not only between families, but within families. I've seen 20-year range in a family, between family members. It has a wide age of onset and certainly a wide range of duration. Certainly can be associated with other neurodegenerative conditions too. And then here's just a picture of the progranulin gene. And as you can see, the changes that occur all throughout the gene. Okay. Next is microtubule-associated protein tau, and Dr. Dickerson mentioned tau. MAPT is what we call that. It's also housed on chromosome number 17. That's just a coincidence. Certainly have found lots of different changes in the gene. They're clustered within this geographic area of the gene. It must be more prone or more to changes or more detrimental. We know that normal tau promotes normal neuronal function and integrity. And so when we lose it, we have these aggregates and tangles that disrupts the cell function and leads to cell death. Altered tau, as Dr. Dickerson mentioned also, is seen in other neurodegenerative conditions. It accounts, again, for a proportion of all FTD, maybe a quarter of familial. Again, if you have a change in this gene, you just about have a 100% chance for having FTD. It's variable also. See lots of behavioral changes, movement changes, and limited to when we see a change in the gene, what we think a person's symptoms will be. Okay. And there's a picture, and it's just showing you a clustering of the changes that occur in the gene. And lastly, chromosome 9 open reading frame 72, or C9orf. The folks who identify a gene get to name it. So the folks who identified this gene came up with this name. This is housed on chromosome 9, its geographic location. Not exactly 100% sure what this gene does, but it may regulate trafficking, transportation of substances within cells, and orderly destruction of cell and cellular components in neuronal cells. And there's this repeat in the gene, and unfortunately it's expanded in folks who have FTD, and the curious factor is this also can cause ALS. So it really was confusing for us all as we were trying to do research. And normally we have about 20 of these repeats, and folks who have FTD or ALS or both have greater than 30. Okay. So we still have a lot of work left to do that we're trying to. Oh, lastly, I'm sorry, just again, the C9 seems to be, the more we know about it, this is just within the last few years, it's accounting for a lot of cases of not only sporadic and familial FTD, but a lot of cases of ALS. So we still have much work left to do, and part of the things that we do is provide genetic counseling. I take a history, medical and a family history. As you can see, genetic testing is imperfect, so try to determine who this might provide information for. Who do you test? Why? For someone who's symptomatic, it's to help contribute to the diagnosis of their disease. Historically, the only way to know for sure what neurodegenerative condition folks have is autopsy or genetics. For folks who are asymptomatic, why would you test? There's not anything we can do about it currently, but hopefully soon. Test results are positive. That means you have an alteration. Negative, you do not, or uninterpretable. We are still the unexplored frontier, genetics. So there are information that we get and we cannot interpret accurately, but we're working on it. The disease is very variable, so even if we find an alteration in a person, we won't be able to tell them when will be the onset, what type of symptoms you will have. And there's certainly psychological impact that goes along with this, not only for folks who are symptomatic. Genetics, we know we have a ripple effect on families. It's not only affecting the person that's sitting in front of me, what I tell them. And we certainly wouldn't want anybody to be discriminated against, although they're not supposed to. That doesn't mean it doesn't happen. So this is a nice continuing of research, and as Dr. Dickerson mentioned, our goal is to return things to our families. So research starts with patients and families. We have a disease such as FTD. We want to learn what the symptoms are. We try to identify not only the genes that cause it, but genes that modify it. Some people have earlier or later onset. Then we test our models in animals, and we develop drugs, and we'll return them to the family. So this is something that we think of as a circular event. It all starts and ends with all of you, and we couldn't do this without your help. So it's really appreciated that you're willing to help and participate. We're trying to identify new genes that cause FTD, factors that modify the disease symptom, and as Dr. Dickerson mentioned, we are the ones that are creating these stem cells. If you think back to that picture that I showed you of a human being and how we make different cell types, we can take a skin cell and erase its memory and just push it down the path to become a neuron. So we actually have FTD neurons in the laboratory that we're studying, trying to understand better, and what's the process that goes on? Is there an earlier way to diagnose people or know what's happening? And then hopefully we can develop some new substances and use them right on our FTD neurons in the laboratory. Okay, and that's it. Thank you.